So it's going to be a bit of a different talk. First of all, I work for university, and I don't do much translational work. It's what it was, before there was a question. This is perhaps an example or kind of blue sky, which we don't know if it's going to be relevant for human health, but certainly it's going to hopefully increase our knowledge regarding a very important molecule, which is RNA. I'm always been interested in understanding how the nervous system develops. And for various reasons. First of all, because just in general, even when I started my medical school, the first things that I really decided that I would have been working on the brain, because I thought it was a very fascinating field of research. But also because it's really interesting to try to understand how you go from a very fairly homogeneous number of neuronal precursors, which now we know they're not exactly identical, but they're very similar in terms of transcriptomics and proteomics, how these fairly homogeneous precursors then differentiate in a very huge, very large number of neurons that are distinct both morphologically and functionally. And this, for example, that can be a Purkinje cells in the cerebellum which control the fine movement, and this could be, for example, a motoneuron with a very long axons. Eventually, all these cells have to make contact to each other in a very precise manner through synapses. And that's really important because the formation of this circuitry between neurons is what underlies all behavior. And we also know when this circuitry goes wrong, we have all series of disorders that we know, for example, can give rise to disorders in the adult. If we think about schizophrenia or autism is more obviously a childhood disorder, but even now we are thinking about even some dementias might start during development when some of this wiring goes wrong. So in the lab, we work on both neurons from the central nervous system, but also for the peripheral nervous system. And in this particular case, I will tell you about some work that we've done on peripheral neurons. These are specifically, they're called sympathetic neurons, and it regulates all your, for example, heartbeat, heart frequency, and all the gastrointestinal function and so on. So they're extremely important, but they are autonomic neurons, so they cannot be controlled by your will. The reason why I'm particularly interested in working on these neurons is because they are quite unique in terms, like as a model system, they're very interesting. First of all, because they, as I say, they innervate all your internal organs, so they're very important functionally. And also because they can be used as a model system very efficiently because they depend completely on a neurotrophic factor, which is called NGF, for growth and survival. So when we deprive these neurons, either in vivo or in vitro, in culture from the NGF, they undergo to massive cell death. So that makes very interesting to try to understand how this factor, this unique factor, can regulate the entire well-being of a neuron. And the second thing is because when I start working on this, it was also very interesting to understand how this neurotrophic factor works. And for people that are interested in more translational aspect, they might recognize that, for example, NGF and BDNF has been considered as potential therapeutic molecules to be used, for example, in dementia, especially the NGF. BDNF has been involved also in Rett syndrome and so on, a series of neurological disorders. And these are molecules, they belong to a family. In reality, they are not very similar, NGF, BDNF, NT3, and NT4/5, but they all bind to a family of tyrosine kinase receptor, which are TRAC. TRAC receptor, TRAC-A, TRAC-B, and TRAC-C. They also bind to another receptors with lower affinity, which is called P75. However, I will not tell you much about what happened in terms of signaling, although we have done some work on that. But rather I wanted to tell you about something that it depends from NGF, of course, but is common to all neurons, also the ones that are not neurotrophic independent. So when I started my lab a few years ago now, I was really intrigued by really the morphology of especially peripheral neurons. So as you can see, these neurons, for example, sensory neurons or sympathetic neurons or motor neurons, they have the cell bodies that is located centrally, either like in paravertebral ganglia or in the spinal cord, the anterior horn of the spinal cord. And then they have to project these very, very long axons during development. And these axons obviously can be literally meters long if you think a giraffe or an elephant, right? So you have this very, very long axon. And there were some studies showing that at least for some protein, the half-life of the protein wouldn't be long enough for the protein to be transported from the cell bodies, where at that time were the only area where ribosomes were found, to the tip of the axons, for example. And here it's very important because a lot of action takes place here. For example, here you have NGF, which is target-derived, so it's released just from the target. But you also have all these important axon guidance molecules. And what these axon guidance molecules do is to tell an axon where to turn in order to reach the proper organs that have to be innervated by the specific axons. So many years ago now, we decided to take a bit of this approach, and we wanted to look, maybe I should introduce first. So one possibility is that instead of the protein, is the RNA that is transported and is translated in the distal part of the axons and in the growth cone. But at that time, when I started working on it, there was very few examples of RNA that could be transported in axons and translated in axons, although many more had been already found in dendrites. And the reason, because there was a lot of skepticism, is because it was very hard for many years to find ribosomes in axons. And of course, if you don't have ribosomes, you wonder, why do you want to really have RNA there? But now we know that they are, actually, thanks mostly to the work from Kristen Holt, I have to say. So why RNA is interesting? So RNA is actually, I think it suffered a lot from the famous dogma theory that was postulated by Watson and Crick, which they say that RNA basically was just transferring information from the DNA to the protein. In reality, so for many years, I think it's been a bit neglected. But then when I was writing this review, I went back and I found these very interesting papers from Czech, which was actually, they were reviews that discussed the origin of life. And the theory was that in reality, the first molecule, so life, originated from this prebiotic RNA. And this prebiotic RNA was called primordial ribozymes. And it has a unique feature. And the reason why it was postulated that could be actually the initial molecules that originated life, because it is quite unique. And actually, RNA is still like this, even in modern cells. It's the only molecules that can combine information storage in the nucleotide sequence, for example, with active enzymatic domain, like, for example, we see in ribozyme. However, I would say that so far we have had a quite sort of clear distinction in terms of class of RNA. You would have the messenger RNA, which is the ones that until not too long ago was considered to be the only really important one, because what encode for protein would be translated into protein. So if you see an mRNA, you assume that this will be translated into a protein. And then there will be the other that have an coding independent function. And obviously, there's been a huge interest that has been grown in the past decade or so, when microRNA, long non-coding RNA, we already knew about tRNA, of course, have been found. And now we know that there are all these other RNA which have coding independent function. But so far, I would say that mRNA has always been considered just a way for translated DNA information into protein. So I just want to introduce a bit more of the RNA, why we are particularly interested in one particular part of the RNA. So the coding seed, the open reading frame for mRNA, as I said, is the area where there are the exons and where the protein is translated from. But then usually it doesn't really, this coding, this open reading frame doesn't even account for even the majority of the RNA, because most RNA comprise a very long 5 prime, actually 5 prime a bit shorter, but very long and very complex 3' UTR. These are untranslated regions, so regions that are not translated, but they are there for a reason, obviously. And in particular, the 3' UTR is particularly interesting for what we wanted to try to understand, which was to see how RNA are transported over long distances and translated at the very tip of the axons. Because there are the RNA, as I say, these are variable length, but usually they're quite long, especially in the nervous system, in neurons. So these 3' UTR can be very long. They form secondary structures with this stem loop, and they act as a docking sites, a bit like what has been described before for the DNA, the docking sites for a series of RNA binding proteins. And these RNA binding proteins are responsible, for example, for the transport, for maintaining the RNA translationally repressed while in transit, or there are microRNA binding sites. That means that when the microRNA is expressed, the RNA will be targeted and degraded. So these are very important. These are all the different things that have been shown. That is like, RNA stability is mostly the 5 prime, but localization, local protein synthesis, translational control is mostly on the 3' UTR. Now we go to another level of complication, if you think about it. Recently, thanks to the advances in genome sequencing and RNA-seq, of course, all the transcriptome has been defined in neurons, in all cells. And in particular, there's been a lot of efforts also on trying to understand how many isoforms can originate from a gene. And this is what was very interesting, was maybe a bit unexpected, is that the most alternative isoforms that you find in any cells doesn't really come from the splicing of one exons in and out, but it comes from different 3' UTR. So it means that this 3' UTR, so even when you have the identical 5' UTR, the identical open reading frame, you can have 3, 4, even 5 different 3' UTR of different length. And that was a bit maybe unusual, but was not unusual, unexpected, I would say. Then when we went through like all these are data obviously taken from many different labs in different sort of cell types, it was noticed that a vast majority of genes contains more than polyadenylation sites, and they can generate RNA with different 3' UTR, and it was also very important to see that the 3' UTR becomes longer, how the neuron differentiates, for example. Also in more differentiated cells, the 3' UTR tends to be longer. And it also was interesting to see that neurons by far express isoform with the longest 3' UTR. But it wasn't clear. Now we are trying to understand why is this. So this something was interesting. And so we took a sort of a brute, force approach in terms of like how to understand the transcriptome that was present just in this distal part of the axons. And we use this model system, which is quite beautiful, not very simple, very labor intensive, in which we grow neurons in these chambers, in which we can isolate just the axons without cell bodies. And then we can isolate the RNA, and then we perform a 3' end seq. So we amplified first and then sequence just the 3' UTR. In this way, we not only knew which are the transcript, the gene whose transcript is in the axons, but also which are the isoforms, and there was any difference in these isoforms. And I won't tell you much about the results of that specific screen. It's a different paper that is currently submitted. But I will tell you just that we got a lot of very interesting data, and we found that the RNA that are transported in axons have a longer 3' UTR. They're more complex, and they're also, these 3' UTR undergo processing in axons. But one thing that we found, at that point we had all these genes, as all the people that work in transcriptomics here, they know that it sounds great when you write a grant, but then you get your results back and you have tons of genes, and where do you start? So there are two options. You either go for the ones that is more fashionable, so it's going to get into a good journal, or you can go for the ones that makes more sense for a reason or for another, which might not be necessarily particularly fashionable. In this particular case, because we were looking at mechanism, I really didn't care much about the function of the genes, to be honest. I want to understand how a neuron would decide, okay, this RNA will go to dendrites, this RNA will go to axons, this RNA will stay here around the cell, in the cell bodies. I thought that was important, it was interesting. I wanted to understand the mechanism, the signaling that would regulate this localization. So I went for the first on the list, which was this not very glamorous name, TP53-imp2. Didn't have anything to do with TP53. It was just found in a screen that happened to be relevant for TP53. So this TP53-imp2 was extremely abundant in axons of sympathetic neurons, really abundant. He had an extremely long 3' UTR, 3.2 kb, which is quite almost unheard of. Usually even in the nervous system, neurons are about average 800 nucleotides. This was 3.2. And it was highly enriched, and it was also highly enriched compared to cell bodies. And this is just like the bird view of the RNA-seq, in which you see that it seems to be just one isoform, very long, which is present in both cell bodies and axons. But it's highly enriched in axons. Okay, when we started looking into this, nothing was known until this lab, and also other labs, I have to say, after this, Zorzano in Barcelona, published this paper linking TP53-imp2 with autophagy. And autophagy obviously was very interesting for us because autophagy is known to be very important for axon growth. So if you have this protein that is synthesized locally and then is somewhat increased autophagy, that would just make sense. So I thought, oh, well, maybe it wasn't such a bad choice. And in collaboration with the lab of Reggie Curovilla at Johns Hopkins University, we made a knockout, a conditional knockout, in which we knock out this gene just in sympathetic neurons. And we observed these are sympathetic neurons at different developmental stages. And as you can see, we did see two important phenotypes, which relate with the fact that these neurons were not responding very well to NGF because there was more cells dying, especially around the P0, so the time of birth. But also, this is a whole mount staining of the heart for sympathetic innervation, is a TH whole mount staining. We saw a decrease in innervation of organs that are targeted by the sympathetic nervous system. Okay, so we had an interesting phenotype, but then we started having trouble, because we got a lot of antibodies, including ABCAM, and we got antibodies by Zorzano. We made 2 antibodies here at the LMCB at UCL. Reggie at Hopkins made other 2, and we could never see the protein. We could see the TP50 if we took the open reading frame and we overexpress it on a normal vector, just 5' and 3' UTR standard, beautiful. And it was also specific, the antibody, because when we use shRNA, the band disappeared, but never at the level of the endogenous. And at the end, after having tried, this is just like 2 years of work, which I'm synthesizing in one slide. At the end, we decided to go for the very, ultimate experiments. We did mass spec from axons, which wasn't really easy, and targeted mass spec, and indeed, we didn't find any. The only thing that we found, a very small peak in cell bodies, but we think this is due to non-neuronal cells, because in order to have enough axons, we had to use explants, and in the explants, the center of the explants in the ganglia, is almost impossible to get rid of all the non-glial cells, the non-neuronal cells. Okay, so tons of RNA, no protein. This is a messenger RNA, is clearly translated, can be translated. So what's going on? So at this point, we decided there must be something that has to do with the 3' UTR. And so Hamish made the sequential deletion of the 3' UTR. And as you can see, when we chopped off, by chopping out different section of the 3' UTR, we would increase the level of translation. So on the right there is a GFP. This is a 3' UTR cloned on the GFP, and you see that clearly you can see the expression. And we also shown that these are just that this was significant. So the shorter the 3' UTR, the better is the translation. And I have to say I don't have the data because we got it yesterday for the revision of this paper, and basically what we found is that in cells like HeLa cells that express the protein, there is an isoform which is pretty much the same length, 1.2 kilobases, the 3' UTR. So clearly this is what's going on. So if you have a short 3' UTR, an mRNA translates the protein. If you have a long 3' UTR, what do? So there is just RNA, there is no protein. What is this mRNA doing in axon? And what we found, I thought was very exciting. I don't know how many of you are familiar with the neurotrophin field. This is a very mature field and pretty much has been done and said, and all knockout has been analyzed to death. So it is it's a field that obviously has been around for many years. And so there are many, almost everything is known regarding how neurotrophin works. But what we found is that these RNA, TP53, interact with TRK receptor directly. This is a RNA immunoprecipitation. And we found that when we knock out, when we use animals that had, well, neurons in which TP53 has been knocked out, so there is not TP53 RNA, we affected all the signaling, TRK phosphorylation, ERK phosphorylation, AKT phosphorylation. And these are the quantification. So the interaction of this RNA with TRK is necessary for TRK signaling. And in data that I will not show you today, we also found that these affect the internalization of the receptor, which is known to be necessary for supporting survival. Okay, but one thing that you always, when I gave this talk around, everybody would say, well, how do you know that there is now very little protein, and that you can't detect it, that the antibody is not good, and there is, you know, the proteasome and this and that, so how do you know? Well, I say the only way we can know it, if we take the RNA, we mutate all the translational start sites, true or potential. We mutated all, there are 4 of them, and then we use this RNA to rescue the phenotype of the mouse. So because if we can rescue the phenotype just with the RNA, when we know that it's impossible just to make protein, so that means that that's a function, is an mRNA that works in a coding-independent function. And these are just the experiments that we did to show that this RNA cannot be translated. And indeed, I want to please focus just on the bottom part of the slides. And as you can see, so when we take neurons and we, with mice that have been knocked out, the TP53 has been knocked out, when we infected with a vector which express the ATG null TP53, we can rescue completely, almost completely, the axon growth. So that means that you know, in this particular case, in neurons, you don't need a protein. So why I think this is interesting, behind the fact that you know, it's a new way of modulating NGF TRK signaling, which is really important actually for it's because this should be seen in the context of other 3 papers, really, 4 papers, 3 findings that have been done, they've been made recently. And because I also think, I think the provocative part is that you'll never look at the mRNA in the same way again. Because when you look at the mRNA, you look just and think, oh, this is important because it makes this protein. But in reality, for example, Christine Holt has published this beautiful Cell paper in which she has mapped all the RNA expression in all different stages in axons. And what she found that I thought was very interesting, that she did transcriptomic and proteomics, and she found a large number of RNA that are present when there is no protein. They are stored there. Is it possible? You know, the RNA is not you know, an eternal molecule, so undergo degradation, has to be resynthesized. So I was wondering if maybe this is just the tip of the iceberg, and maybe RNA that they're there in the absence of the protein are working in a completely different way. And then eventually, when become translated, they might change their function. Another implication of these findings is that really, in our case, we have shown that when the RNA is translated, it is important, is involved in regulating autophagy. But when it's not, it does completely different things, and most likely what it does is regulating protein-protein interaction maybe, or protein receptor interaction, formation of the endosomes, completely different. And these are the three cases I want to say that are very exciting because they're very important finding, very important discovery. This is one, you know, the last one is ours, so I'm excited because it's ours. But the other two are very interesting. One is from the laboratory of Gerhard Schratt, and he showed something that was slightly different. But he showed that when an exon of this UBE3A, which is an ubiquitin ligase, this is all in neurons, of course, when it's spliced out, the RNA is not translated, and it works almost like a sink for microRNA, so in a completely translation-independent manner. These are even more exciting because ARC genes for neurobiologists has been one of the most important genes for various reasons, especially if you study synaptic activity-dependent gene expression. This was cloned when I was at Hopkins in Paul Worley's lab, and this is genes that quite dramatically increase, very dramatically increase in response to synaptic activation. In hippocampal cortical neurons, it goes up. And it's been described as important for cytoskeleton and other things at the synapses and so on. This is also transported at the synapses and translated at the synapses in response to synaptic activation. So it's a very interesting gene. But what the Vivian Budnek lab and Shepard lab in two independent studies shown is that the ARC, the same RNA, can work in two ways. So ARC was known to have this GAG domain, which are very similar to HIV virus. And what they found is that the ARC can be translated into protein, and the ARC molecules, they made a sort of like a capsid, really a capsid. If you see EM, it looks like a virus, really. And inside this capsid-like structure, there is ARC RNA. And what they found in Drosophila, that this virus-like structure can pass the neuromuscular junction and basically transfer the RNA from one cell to another. This way you have ARC is even better because you have in the same cells, in the same structure, you have a coding-dependent and a coding-independent function, which completely obviously have changed the way we have to think about these molecules and what it does really. And this is what we found for TP53 IMP2. Okay, and now I want to thank the two people that did everything. I have to say, Hamish was a PhD student. He's finished now. He's about to move on, and he really was very, very brave because when he came, we had spent almost 3 years trying to make these things to work, and then I had given up completely because we couldn't see the protein. So I said, well, there's no point. And he was very brave. He said, I'm going to see if I can find out what it is. And Katia Andreassi, which is the person that works on RNA in my lab. And these are several collaborators, some from the Creek. Marco Gaspari did all the mass spectrometry in targeted mass spectrometry. Adolfo and Anne Willis helped us with and I have to thank also my funding buddies. And if you have any question, I'll be happy to answer.